Hello, welcome to Biology, the scientific study of life. The cell cycle clock, cyclins and cycle-dependent kinases. Rhythmic fluctuations in the abundance and activity of cell cycle control molecules pace the sequential events of the cell cycle. These regulatory molecules are proteins of two main types, kinases and cyclins. Protein kinases are enzymes that activate or inactivate other proteins by phosphorylating them. Particular protein kinases give the go ahead signals and the G1 and G2 checkpoints. The kinases that drive the cell cycle are actually present at a constant concentration in the growing cell, but much of the time they are in an inactive form. To be active, such a kinase must be attached to a cyclin, a protein that gets its name from its cyclically fluctuating concentration in the cell. Because of this requirement, these kinases are called cyclin-dependent kinases or CDKs. The activity of a CDKs rises and falls with changes in the concentration of its cyclin partners. Notice this figure molecular control of the cell cycle at the G2 checkpoint shows a fluctuating activity of the cyclin CDK complex that was discovered first, called MBF. Note that the peaks of MBF activity correspond to the peaks of cyclin concentration. The cyclin, notice that the cyclin level rises. Notice during the S and G2. Notice and S and G2 phases, then falls abruptly during mitosis M. Notice that the initials here also. The initial MPF stand for maturation promoting factor, but we can think of MPF as M phase or promoting factor because it triggers the cell's passage past the G2 checkpoint into M phase. When cyclins that accumulates during G2 associate with CDK molecules, the resulting MPF complex initiates a mitosis, phosphorylating a variety of proteins. MPF acts both directly as a kinase and indirectly by activating other kinases. For example, MPF causes phosphorylation of various proteins of the nuclear lamina, which promotes fragmentation of the nuclear envelope during prometaphase of mitosis. There is also evidence that MPF contributes to molecular events required for chromosome condensation, for spindle formation during prophase. During anaphase, MPF helps switch itself off by initiating a process that leads to the destruction of its own cyclin. The non-cyclin part of MPF, or the CDK, persists in the cell in inactive form until it associates with the new cyclin molecules synthesized during the S and G2 phases of the next round of the cycle. What about the G1 checkpoints? Recent research suggests that the involvement of a least 3 CDK proteins and several different cyclins. At this checkpoint, the fluctuating activities of different cyclin CDK complexes seem to control all the stages of the cell cycle. Bon. Again, fluctuation of MPF activity and cyclin concentration during the cell cycle. Notice these are represent relative concentration, MPF activity, this one cyclin, and notice that G1S, G2M phases. Also notice also the concentration here. Another figure represents molecular mechanisms that help regulate this cycle. One represents synthesis of cyclin begins with inlet S phase and continues through G2 because cyclin is protected from degradation. During this stage it accumulates. Notice 2 CDK accumulated cyclin molecules combine with cycle CDK molecules, producing enough molecules of MPF to pass the G2 checkpoint and initiate the events of mitosis. 3 here, MPF promotes mitosis by phosphorylating various protein. MPF's activity peaks during metaphase. Stage 4, during anaphase, the cyclin component of MPF is degraded but terminating the M phase, the cell enters G1 and G1 phase. These are steps of the cell cycle are timed by rhythmic fluctuations in the activity of cyclin-dependent kinases, CDKs. Here we focus on a cyclin CDK complex called MBF, which acts at the G2 checkpoint as a go-ahead signal triggering the events of mitosis. Stop and go sign, internal and external signals at the checkpoint. Research scientists are only in the early stages of working out the signaling pathways that link cyclin-dependent kinases to other molecules and events inside and outside the cell. For example, they know that the general active CDKs function by phosphorylating substrate proteins that affect particular steps in the cell cycle. In many cases, though, scientists don't yet know what the various CDKs actually do. However, they have identified some steps of the signaling pathways that convey information to the cell cycle machinery. And complex of an internal signal occurs at the M phase checkpoint. Anaphase, the separation of sister chromatids, does not begin until all the chromosomes are properly attached to the spindle at the metaphase plate. Researchers have learned that kinetochores not yet attached to spindle microtubules send a molecular signal that causes the sister chromatids to remain together, delaying anaphase only when the kinetochore of all chromosomes are attached to the spindle will the sister chromatids separate owing to activation of the proteins holding them together. This mechanism ensures that daughter cells do not end up with missing or extra chromosomes. By growing animals cells in culture, researchers have been able to identify many external factors, both chemical and physical, that can influence cell division. For example, cells fail to divide of an essential nutrients. It is left out of the culture medium. This analogs to trying to run an automatic washing machine without the water supply hooked up. And even if all other conditions are favorable, most types of mammalian cells divide in culture only if the growth medium includes specific growth factors. Growth factor is a protein released by certain cells that stimulates other cells to divide, called a growth factor, for historical narrowly called mitogen. One such growth factor is a platelet derived growth factor, short-term PDGF, which is made by blood cells called platelets. This experiment illustrated in this figure, notice that, demonstrates that PDGF is required for the division of fibroblasts. In culture, fibroblasts, a type of connective tissue cell, have PDGF, notice that, molecules to these receptors, which are receptor tyrosine kinases. It triggers a signal transduction pathway that allows the cells to pass the G1 checkpoint and divide. PDGF stimulates fibroblast division not only in the artificial conditions of cell culture but in an animal's body as well. When an injury occurs, platelet release PDGF in the vicinity, the resulting proliferation of fibroblasts help heal the wound. Researchers have discovered at least 50 different growth factors that can trigger cells to divide. Different cell types respond specifically to a certain growth factor or combination of growth factors. The effect of an external physical factor on cell division is clearly seen in density-dependent inhibition, a phenomenon in which crowded cells stop dividing. As first observed many years ago, cultured cells normally divide until they form a single layer of cells on the inner surface of the culture container, at which point the cells stop dividing. If some cells are removed, those bordering the open space begin dividing again and continue until the vacancy is filled. It is originally thought that a cell's physical contact with neighboring cells signaling it to stop dividing. However, while physical contact may have some influence, it turns out that the amount of required growth factors and nutrients available to each cell has a more important effect. Apparently, when a cell population reaches a certain density, the availability of nutrients becomes insufficient to allow continued cell growth and division. Boom. Most animal cells also exhibit encourage dependence to divide. They must be attached to a substratum, such as the inside of a culture jar or the extracellular matrix of a tissue. Experiments suggest that encourage is signaled to the cell cycle control system via pathways involving plasma to the cell's plasma membrane proteins and elements of the cytoskeleton linked to them. Density-dependent inhibition and encourage dependence appear to function in the body's tissues as well as in cell culture. Checking the growth of cells at some optimal density and location. Cancer cells, which we discuss next, exhibit neither density-dependent inhibition nor encourage dependence. Back to research. The name of experiment, notice that the steps, results, And conclusion, the research called does platelet-derived growth factor BDGF stimulates the division of human fibroblast cells in a culture experiment? Notice that a symbol of connection tissue was cut up into small pieces. Notice here, enzymes were used to digest the extracellular matrix, resulting in a suspension of free fibroblast cells. Notice, and cells were transferred to sterile culture vessels containing a basic growth medium consisting of glucose, amino acids, salts, and antibiotics as a precaution against bacterial growth. BDGF was added to the half of vessels. The culture vessels were incubated at 37 Celsius degrees. Results in a basic growth medium without BDGF. The control cells failed to divide. Also notice that the basic growth medium plus BDGF cells proliferated. The CM shows cultured fibroblasts. Conclusion, the experiment confirmed that BDGF stimulates the division of human fibroblast cells in culture. Okay, thanks for listening.